We owe almost all of modern genetic testing to a bacteria that lives in hydrothermal vent. In order to look at something's genetic sequence, you need replication enzymes to amplify the amount of DNA present so that it's detectable. First, you have to heat the DNA so that it splits open from its double helix into two single strands. And this is done at a very high temperature, over 90 degrees Celsius. Then, the temperature of your reaction is taken through a few different steps for different lengths of time in order to allow the anchors to attach and the enzymes to extend. This happens over and over, usually 25 to 35 times, in a process called polymerase chain reaction, or PCR. The problem is that initial heating step destroys the enzymes that are used in the final steps. I've said before that enzymes and proteins require specific temperatures in order to fold properly and be effective, and most species only have proteins and enzymes inside them that operate at their body temperature. So where do you find an organism that's adapted to survive at 95 degrees Celsius? In hydrothermal vents, of course. Enter Thermus aquaticus, a bacteria discovered in 1969 in the Mushroom Spring in Yellowstone National Park. Originally, PCR was very slow and done by hand. You moved your tray of tubes from each temperature block set at a different temperature, and after every heating step at the beginning, you had to add fresh enzyme for the extension step. Needless to say, this was very painstaking and tedious for a process that can take hours to run. In 1986, Randall Sakey built on the PCR technique that had been invented in 1983 by his co-worker Kary Mullis. Instead of using human DNA polymerase, he used the polymerase from Thermus aquaticus. This allowed the process to be automated and to be done much faster. Today, we have Taq polymerase mixtures in one tube that contain everything you need for the entire reaction. And we have single small heating blocks that handle all the temperature changes automatically. So all you do is you add your Taq and your DNA to your tubes, and then you set it in the machine, you hit start, and then you come back a couple hours later and it's done. Mullis was awarded the 1993 Nobel Prize in Chemistry for the invention of PCR, and Sakey was left out entirely. But at least that means he also didn't come down with the Nobel disease later on in his career like Mullis did.